We often think of DNA as a stable blueprint of life, passed down unchanged from generation to generation. But this is not entirely accurate. DNA is surprisingly dynamic, constantly undergoing changes and rearrangements. These changes, while often subtle, can have profound effects on the evolution of life. One of the most dramatic examples of this is polyploidization. Polyploidization is a process that doubles the number of chromosomes in an organism's cells. This might sound like a recipe for disaster, but surprisingly, many plants and some animals tolerate it quite well. In fact, polyploidization is a significant driver of evolution, particularly in plants. Imagine a plant suddenly having twice the genetic material. Polyploidy is a fascinating phenomenon where organisms have more than two sets of chromosomes. This abundance of genes can lead to new traits and adaptations. With extra copies of genes, plants can experiment. These additional gene copies provide a genetic playground for evolution. Some copies might change and develop new functions, while others keep their original roles. This can lead to a variety of new traits that can be beneficial for survival. This genetic flexibility allows polyploid plants to adapt to new environments and evolve into distinct species. They can thrive in conditions where their diploid relatives might struggle. Many of our crop plants, like wheat and potatoes, are the result of ancient polyploidization events. These events have given us the diverse and resilient crops that are essential for our food supply today. Section 4. Copy and paste genes. Duplication and new functions. Polyploidization isn't the only way genomes change. In fact, the dynamic nature of genomes allows for a variety of mechanisms to introduce genetic diversity. Sometimes just a single gene or a small group of genes gets duplicated. This process can occur due to errors during DNA replication or through the action of mobile genetic elements. These extra gene copies are free to mutate and evolve new functions, potentially benefiting the organism. For instance, a duplicated gene might acquire a mutation that allows it to perform a new advantageous role. Over time, these duplicated genes can give rise to entire gene families, each specializing in a particular task. This specialization can lead to increased complexity and adaptability within an organism, contributing to the rich biodiversity we observe in nature. Section 5. Jumping genes, transposons, and their genomic adventures. And then there are jumping genes or transposons. These fascinating elements of our DNA are like tiny nomads, constantly on the move. These are DNA sequences that can move around the genome, inserting themselves in new locations. This movement can happen through a cut and paste mechanism or a copy and paste mechanism, allowing them to proliferate within the genome. While they might sound disruptive, they actually contribute to genetic diversity. By inserting themselves in different parts of the genome, they can create new genetic combinations and variations. Transposons can alter gene expression, create new mutations, and even shuffle large chunks of DNA. This shuffling can lead to new traits and adaptations, which can be beneficial for the survival of a species. In conclusion, the genome is far from static. It's a dynamic entity, constantly evolving and adapting. From entire genome duplications to the subtle movements of transposons, our DNA is in constant flux. This constant change is a fundamental aspect of life. This dynamic nature is a key driver of evolution, leading to the incredible diversity of life we see today. The ever-changing genome is a testament to the resilience and adaptability of life on Earth.